This is Dr. Dave Seftel reporting for Macular News, and we're here with Professor Margaret DeAngelis from the Moran Eye Center at the University of Utah. Welcome, Professor.
Thank you. Hello.
So tell us a little bit about the presentation that you made at the Harvard AMD conference today.
The presentation I made was to show how genomics, or genetic association between age-related macular degeneration, can translate into biological meaning so that we can get to disease mechanism. And that's what I talked about.
And you looked at a whole host of genes and their association. Tell us a little bit about that.
I looked, a number of genes have been reported, particularly by a number of authors. A number of people spent many years, particularly that are part of the AMD Gene Consortium, which is an international consortium by many people, and it's sponsored by the National Eye Institute. And we've uncovered many genes by many cohorts throughout the world. And I wanted to show how important those genes are at the expression level. Are they different between those with disease than those without disease? And looking at the intermediate form of AMD and the type that's the wet form of AMD or the advanced form of AMD, we showed that none of those genes change in their messenger RNA level. Now, that doesn't mean that they're still not important. It just means that they don't change at the messenger RNA level. And gene expression is thought of as the first step when we look at biological function. So that's what I showed today. And then another thing we showed that was important is there's two tissues within the eye that are affected by age-related macular degeneration. One of those is the retina, which is the camera of your eye, and the other tissue is the retinopigment epithelium, which is the supporting structure of your rods and cones. And that piece of tissue, the retinopigment epithelium, which is a single layer of cells, appears to be the most important and the most severely affected at the messenger RNA level in age-related macular degeneration.
Tell us, Professor, what was the association between the RPE and certain genes?
The association between the RPE and certain genes is that we showed that inflammatory pathways that are involved in diseases like Alzheimer's disease, are important to the intermediate form of age-related macular degeneration. And people who have the intermediate form of age-related macular degeneration are at higher risk of going on to have the more advanced forms, like the wet form of age-related macular degeneration. And although there are therapies for the advanced form of age-related macular degeneration, those are limited in their applicability, meaning that not everybody who gets them responds to them, and they don't reverse your loss of vision over the long term. So we showed that some of the pathways that are intermediate, that are responsible for the intermediate, overlap with the advanced form, but some of them are unique to each form. And right now we're trying to confirm those novel genes involved in those pathways, because they weren't any of the genes that have been shown by genetic association studies. And they may represent novel therapeutic targets, and that's what we're hoping for.
That's the next question. What are the translational implications of your work?
Well, I hope they're important because this is a disease that runs in my family, hence why I'm passionate about finding a therapeutic target. So the translational implications is that if we can confirm some of these genes that we have found as a first step in the biological implications, is that we'd like to go after the pathways they function in. And when we look at the pathways, we want to look upstream of what they function in, because you want to go after something that's going to target many genes at the same time, but be focused or localized to the eye, not have toxic side effects to the rest of the body, and then hopefully produce an animal model that you can manipulate and work with. And even though we don't have an ideal animal model, if you can work with, say, a mouse, for instance, you can mimic some parts of the disease in the eye of the mouse. That would be ideal.
So we heard in the discussion a question of using genes as targeting for specific individuals. Their therapy would be gene targeted. Do you see that as developing into a viable platform where everybody will be tested and therapies will be gene directed?
Yes, I do. But we also have to take into account people's medical histories, because people's hypertension, their level of cholesterol, their diet and nutrition is going to play a role as well. And particularly their environment, our natural environment, the amount of sunlight we get, the amount of toxins we get, the vitamins we take. And that's a whole field known as epigenetics, and the amount of methylation without changing the sequence to our DNA. And that can affect it. And what occurs in our eye isn't the same as what occurs in our blood. We have the DNA information as well as the mRNA information of each of these patients. Then we want to understand why their genes are going up or down. And we're going to do a whole genome methylation on them. And maybe it could be explained by methylation that's affecting the expression level. And we're going to try to understand that. Another thing we're going to look at, we have serum. So we have protein on each one of these individuals. And we're going to look at their metabolomics and try to understand that. So we're going to try to come at the problem from a few different methodologies and try to pinpoint disease causality.
So tell us a little bit about the gene database in Utah.
Okay. The gene database in Utah it's a wonderful resource. And part of that is because of the people who work on it. It's run by Dr. Ken Smith, who's absolutely a terrific collaborator and wonderful to work it with. But part of this resource is, I've got to give credit to the people who work at the Utah Lions Eye Bank, particularly Wade McIntyre and Chris Hanna, and our wonderful chairman, Dr. Randall Olson. And it's a team effort in creating this with the Utah population and the families who give up their eyes to make this happen, and their time and their efforts in giving up their information to create the Utah Population Database and being able to access families and their information and the genealogies that go back to the 1800s. And it's just a super wealth of information and knowledge. And another thing, it's also a very homogeneous population. So that also reduces the number of false positives, which can skew many genetic association studies and also increase variance within studies that have biological meaning, especially in humans where you see a high degree of variance, unlike inbred mouse strains where you wouldn't see that as much.
Would you like to add anything else specifically about projects and programs that you are currently working on?
Yes, all projects and programs, besides needing wonderful patient participations by the patient and their families, also need funding to succeed. And as NIH money is extremely important, it's not as plentiful as it once was. And once you may get a grant approved, it can take up to a year to get the actual funding. So it's really, really important to get donor money and foundation money, because that can really move the research forward very quickly. And none of this could have been done without private donor monies, and particularly from the Research to Prevent Blindness, the Reeves Foundation, an aging grant from our Aging Institute at the University of Utah, which directly funded the RNA-seq work. But all these organizations really helped to build the donor eye bank, and actually the National Eye Institute helped too to build the donor eye bank. That's our eye bank with our laboratory. So you can always do more with more money. Like the eye bank could be bigger. And I've got to thank my staff that gets up in the middle of the night and helps to collect these postmortem eyes, because timing is everything.
Thank you so much, Professor, and we wish you all the best and the greatest support for your program.
Thank you.